Okay, thank you very much, Andrey. It's really great to be here, and thank you for the invitation. So it has been actually you already invited me once, but I was let's say not ready. I thought okay, I should make some more complete story on organelles, and that now we invited you once again and the second time, and then say okay, that's good, and it's really nice to talk to community on biomembranes. I'm always happy to communicate with this community. I'm from chemistry field, but we love to develop tools that would help membrane biophysicists and in general people who work on cell biology to visualize better what is happening at the cellular level with molecular resolution or with as high resolution as possible. And today I will be talking about tools that we are developing in our lab, and more precisely fluorescent probes for imaging lipid organization in different parts of the cells and organelles. So I also include the cell membranes, cell plasma membranes here. And yeah, and let's go. So first, I would just like to present the team, and our team, Nanochemistry and Bioimaging, works on several directions. We have two main streams, which is fluorescent molecular probes and fluorescent nanoparticles. And within fluorescent molecular probes, there are different directions. One is on biomembranes, and this is what I will be talking today. But I would like to say that we're also working hard on development of probes for nucleic acids, for RNA imaging based on aptamers, and on membrane receptors, so on targeting specific proteins on the surface of the plasma membrane and going from cellular to animal level. And nanoparticles, we are also very interested, and this was in activity since probably 6 or 7 years. We are interested because they are ultra bright and they are mainly oriented towards development of diagnostics systems for ultra sensitive detection of nucleic acids for point of care diagnostics in particular. And but we also interested to use them in bioimaging at the cellular level and the animal level. But I will be talking about this part about biomembranes and organelles. Okay. So cell membrane organization. So you probably have seen millions of times these schemes. So the lateral organization of cell plasma membrane is not that simple, and it is in part because the lipid composition of those membranes is extremely complex, and this can generate structures, lipid structures of different organization. In particular, let's say extreme structures that we can consider model ones like liquid ordered and liquid disordered phases. And the theory of Kai Simons in the late 90s about lipid rafts or lipid microdomains suggested that these two kind of phases coexisted on the surface of the cell. Instead of to replace this mosaic model where the lipids were supposed to passively diffuse and mix between each other. So here it's more about assembly of lipids of a similar kind, forming some sort of rafts of domains that give the control activity of certain proteins. And this theory came from the methodology, which is related to detergent-resistant membranes, which you probably all know very well. Those were structures that was enriched in cholesterol and sphingomyelin. And so from that, the whole story started. Now, the thing is that these rafts are probably, how to say, there were a lot of debates about the existence. And this is in part because methodology to visualize them are not perfect. So we cannot really, with a good time and spatial and temporal resolution, to visualize them in live cells. And so that's why you have numerous techniques that try to address the problem of the lipid organization and presence of those domains. And in particular, so you have a number of methods based on labeling. of those structures using like cholerotoxin, GPI proteins. And then you have tools where you have, so this come from recent reviews of Gardini and colleagues. Then you have tools to study membrane domains using the model membranes like giant physicals. I will be talking about this old method DRM isolation. Then you have microscopy techniques where you can track the molecules in different environments which are more constrained or less constrained. And like this, you can, based on diffusion, you can conclude whether it is ordered or disordered phases or rafts or not rafts. And you have energy transfer methods. You have, of course, mass spectrometry method, which is very interesting. You can also directly you can do imaging with this technique. And I will be more talking about techniques where we use smart probes that change the color. So it's presented here. So these are domain-specific probes. Domain-sensitive probes. So you have, in principle, two types. So you have domain-specific probes, which distribute either in one or another phase. Like this can distinguish them. But then you have domain-sensitive probes, which are those which can really directly address the biophysical properties of the membrane by changing the color of fluorescence lifetime, etc. So you have also specific RAF targeting drugs, especially derivatives of cholesterol, etc. And then, okay, so let's go to the technique which is related to color switching molecules that are sensitive to biophysical properties membranes. And in particular, these are solvatochromic molecules. And the solvatochromic molecules, these are molecules that change the color as a function of polarity. And usually it is a push-pull molecule, which on electronic excitation becomes highly dipolar. And this dipole interacts with the environment. And so in a highly polar environment like water, the dipole will be stabilized, so the energy will go down, and therefore the wavelength maximum will shift towards red. And therefore, when we change organic solvent from apolar to polar, the color changes from blue to red. And so here I show the molecule of prodan, which is actually the core fluorophore flow of that molecule. And this polarity is a fundamental property of all biomembranes, because biomembrane is actually a highly micro-heterogeneous system where on the surface you have environment rather close to water, let's say surface or out the polar head groups, and then in the lipids or chains you have a polarity rather like oil, and therefore this interplay between polarity is actually defining the whole structure and behavior of plasma membranes and its capacity to accommodate proteins, etc. So therefore, what happened when this kind of environment-sensitive, polarity-sensitive molecule binds to the membrane? So here it is in water. When it is in water, it is usually not highly emissive, but it is redshifted. And then when it binds to lipids, especially at the level of chains, fatty acid chains, the polarity drops of environment, and you have a strong blue shift with a strong increase in the fluorescence. And this is a typical behavior of Nile Red. And you will see that this actually can be used for imaging detecting properties of plasma membranes or cell membranes in general. So as a general scheme, the probe is composed of the solvatochromic dye, then the polar head and the chain, and then you simply add the dye to the probe to the cells, and it assembles spontaneously at the place you want. If your targeting group is good, it will target where you want. And then based on fluorescence, especially the color of fluorescence, we can conclude about, or we can decipher the membrane organization, phase state, of course, the structure, etc. Okay, so let's come back to this heterogeneity and the lipid organization of membranes. So you have two types of heterogeneity. On one hand, you have the lateral heterogeneity, which I already mentioned, and then you have also interleaflet or transmembrane heterogeneity, where the outer leaflet is rather rich in bromelain phosphatidylcholine, and then the inner leaflet is rather phosphatidylethanolamine and phosphatidylserine. So it is very important that the probe is located at a given leaflet. And then the lateral heterogeneity here, the disordered phase is rather considered as a loosely packed phase of lipids, is highly dynamic, and therefore we consider it rather polar and hydrated. And then you have the liquid ordered phase of saturated lipids and cholesterol, which is tightly packed. It is dehydrated and apolar. So therefore, if you place a solvatochromic molecule in these two phases, you are supposed to see changes in the color of emission of the probe. And this is what is used in case of Laurdan and DMX, for example, which was mentioned earlier, polarity sensitive probes. But we selected Nile Red. We like Nile Red very much because of its red emission, high photostability, etc. But Nile Red itself does not really stain well membranes, not specifically. If you add it to the cell, you see that it goes inside and you see the dots. This is the droplets, and the rest of the membranes not specifically distributed. So now, if you add, so to do more specific imaging, you need to add a targeting group. And in the early, or let's say already more than 10 years ago, we developed the first prototype of Nile Red that stained specifically cell plasma membrane. In this case, we added sweet-aryanic group with the alkyl chain to the fluorophore, and so it is a lipid-like charged group, and the chain, it looks like lipid. Molecule is sufficiently soluble; it does not precipitate when you add it to the cells, and it goes and binds rapidly to cell membrane, and you see only specific cell membrane staining. And why it is specific? Because it binds only one leaflet. And how we show that? We use sodium dithionate. That was actually a unique property of Nile Red, that you can bleach it instantaneously with sodium dithionate by reducing actually the fluorophore. It's reversible reduction, and it's very fast. And so what we did at that time, we prepared lipid vesicles containing fluorescent dye on both sides, and added sodium dithionate. And what we observed, that we actually bleached only 50% of the dye. And then when we added tritone X100, which kills the membrane that opens up the lipid vesicles, it drops further to 0. So then, okay, we said, then we did the other experiment, and we added the dye from outside, and then added sodium dithionate, and we could see that we had complete bleaching. In case of Nile Red, there was always bleaching was complete. So this experiment showed that, in fact, the dye will be where you put it. So if you put it on both sides and you bleach, it will be on the inner leaflet, and if you add it on the outside, it will remain outside. Flip-flop was very slow. It was like 7% within 24 hours or something. So within 7 hours, it was like 5%. Sorry. So the flip-flop is a very slow process for this probe. Okay. So now we go to imaging, and as the molecule changes the color upon the phases, and this is, you can see upon phase change. For example, if you compare a liquid ordered phase composed of sphingomyelin cholesterol versus DOPC, which is liquid disordered phase, you have a strong blue shift in ordered phase, like 50 nanometers or something. And this shift means the real emission color changes. But for microscopy, it is much easier to study color changes by recording two channels, by splitting with a dichroic mirror into the blue channel and red channel. So you do it splitting somewhere in the middle between these two maxima. And then, like this, in ordered phase, you will see more signal in the blue channel.
Bu kadardı. İzlediğiniz için teşekkür ediyorum. Hoşça kalın.
Yeah. So,
Anlaşma olabilir mi? Doğu
could you please switch off the microphone?
Farklılaşabilir mi? Bunu konuşacağız. Müsaadenizle.
Yeah. Okay. So, and then we record the red channel. It will be rather disordered phase, which will be more visible. So now when we go to imaging of giant vesicles, which are model membranes that can be generated by electroformation method, in liquid disordered phase, you would see a different color from the liquid ordered phase. So the color is actually a pseudo color, which is generated from the, which corresponds to a given ratio of the blue channel to the red channel. And so this, from this color change, you can identify ordered or disordered phase. And then if you do mixed phases, you can see individual domains of different color, and you can directly link this color to the phase in the pure phase. Okay, so then when we use this approach to image cells, our big disappointment was that the cell membrane had a relatively homogeneous distribution of the color, which means we did not really see these domains. It was not at all obvious to see or appearance of some heterogeneity. So, but what we really did, we managed to detect is changing lipid order. And that we did by varying, for example, cholesterol by extraction with methyl beta saccharolistin. In this case, you can observe a nice changes towards decrease in the order, and depend on the time of incubation, for example, the color changes. Okay, so that was the early work. We observed we couldn't observe rafts, we could just generally address the lipid order on the membrane surface. So no rafts. Okay, then can we really study them at high resolution? Because now domains are considered to be much smaller. They are also dynamic. So can we do super resolution? And so probably you all know what is super resolution technique. The techniques, we have different approaches now. And the ones we work a lot are single molecule localization microscopy, where we exploit dark and on and off states of the molecule. And in this case, usually what we do, we turn off majority of molecules, they're in off state, and then we record just a few molecules that appear frame by frame, and then you can localize and put them all together and reconstruct the image. Actually, it can be depicted here. It's a nice video made by Ricardo Henriques. And here you can see that initially you record the individual spikes, and you localize these dots, and then you can reconstruct a nice picture. Okay, but to make blinking solvatochromic probes is not that obvious. They are usually, first, they are not really photostable, and Nile Red doesn't really blink. So to make Nile Red blinking, what we did, we considered a method which is called paint, where actually Nile Red was exploited originally, that uses reversible binding of the molecule to the membrane. As you remember, as I said before, in water it's not emissive, but in lipids it is emissive. So in principle, this binding can be used to have turnover. And so what we did, we actually functionalized, we changed the design of our original probe, NF12S, and we introduced a charged group and a short linker, a short anchor lipophilic chain. In this case, we decreased the affinity of the probe to the membrane, and like this, the molecule could probably have low affinity to the membrane. And we made exactly very similar analog with a long chain, which is supposed to bind irreversibly. And then when we did affinity study in different lipid concentrations, we could see that the probe, which has a short chain, it binds very weakly, which has low affinity, and probe with long chain binds very fast, with high affinity profile. And this you can see in single molecule measurements. So this was done in collaboration with Kik-Soo in Berkeley. So what they did, they deposited a monolayer of lipids on the surface, and then they added the probe at low concentration. And what they could see that there's our best heat molecule. It shows a lot of single molecule events on the surface, even more than the original Nile Red, while the probes with high affinity, with long chain, they show very few events on the surface. So this molecule was actually perfect for doing super resolution imaging where you have on-off switching. But when you just add them to the cells, what you could immediately see that the best probe is actually not the one which binds reversibly, but the one that binds well. So this one is actually the brightest. It was much brighter than our original NF12S. It has much better, more efficient staining. And then NF12A, which has short chain, it has a very weak binding to the cell surface. So we have to amplify 10 times the signal to see something. So this guy with long chain is good for imaging, just for classical confocal imaging like this, reconstructing. But NF12A is for super resolution, and this is where it is powerful. So now you can compare actually reconstructed super resolution imaging paint of this dye, where you have a nice image of the cell, and compared to, for example, NF12S, which binds irreversibly. It just bleaches, and you cannot really reconstruct any super resolution. So first of all, it works in super resolution. Then if we can zoom, we actually could observe lots of different small structures on the membrane surface, like fiber-like structures and the dots. So those dots were endosomes, and these guys is the more tricky. And so what we later did in collaboration with KXU is to do a spectral result paint, where actually the light goes through a prism, and so for each individual molecule, you can reconstruct a fluorescent spectrum, and then, like this, analyze the color response to the lipid order. And what we found out that the membrane surface do not have really homogeneous distribution of color. And in this case, those little dots, which are endosomes or exosomes forming, and also those little fibrous-like structure, they were systematically characterized by red-shifted spectra, which means that this banding leads to a kind of loss of lipid order. And so there, we observed this as a kind of systematic behavior every time the membrane was distorted. We had change in the order. Okay, so now I would change the gears and say that we used also this approach to target specifically plasma membrane, this anchor group, to another molecule. In fact, the reason we did it was Erdinch, who was very much interested in Lordan. He loved Lordan, and he said, "Oh, I also want to have Lordan which targets specifically plasma membrane." And say, "Okay, we are just doing it for Nile Red. Why not we try it for Lordan?" But so we took actually a Prodan unit and coupled it with our Prodan is actually a short chain analog of Lordan, and we coupled it to our preferred anchor group was a sulfonate and alkyl chain. And so like this, by exploiting a solvatochromic property of Prodan, we could distinguish order, disorder, phase by color change. The same like for Nile Red. And when you check it with vesicles, you can clearly see the typical behavior like Lordan in ordered phase, blue shifted with respect to disordered phase. Okay, then the experiments in plasma membrane vesicles, so giant plasma membrane vesicles, which was actually done by Erdinch, we could see clearly that we can distinguish ordered and disordered phases on those reconstructed membranes, and the resolution between these two phases was better. You can see it here than for Lordan or C-Lordan. So C-Lordan, by the way, is show you it's a carboxy derivative of Lordan. Okay, and the motivation, the motivation, I guess, I think I already explained. So we wanted to be more specific to plasma membrane. C-Lordan was an attempt to do it, but it was, as you will see, it's not really so efficient. Okay, so now as we will target specifically plasma membrane by labeling just the outer leaflet, we need to prove that it binds on the outer leaflet. And for Lordan or Prodan derivative, it was much more tricky because, for example, molecules like sodium dithionate salts like that do not really react and do not bleach. So what we use a quenching process in this case. And for that, we need to design a specific quencher based on biologen. And so what we did, we considered two situations. If the molecule binds only to the outer leaflet, it should be efficiently quenched. But if it binds to two leaflets, in this case, quenching will be weak. Weaker, let's say 50%. And this is what we observed. When we compare this Pro12A, which is our leaflet binding probe, we could see a very nice quenching with our Violagen quencher. In case of Laurdan, the quenching was much weaker and was like say half of what we observed for Pro12A. So which confirmed that their binding mode is quite different. And so this is rather one leaflet binder, and Laurdan distributes between both leaflets. So when you add this to the cells, you can clearly see the difference between labeling. So for Pro12A, we observed a much more specific labeling of the plasma membrane, while for Laurdan, it was strong internalization, and it was even stronger for C-Laurdan, which was a surprise. Okay, and then we tested the effect of cholesterol extraction, and we observed quite strong effect of cholesterol extraction of Pro12A and much weaker response of Laurdan, which is probably related to the leaflet specificity of this Pro12A. Okay, so now we go to the main story. So I just wanted to introduce our continuous effort to study membrane surface, but there are also lots of other membranes inside the cells, and they're in endosomes, in endoplasmic reticulum, Golgi, mitochondria, etc. And to target them, and why it's interesting to target them is that their organization, lipid organization, is very different in different organelles. And just an example, the cholesterol, 60% of cholesterol is located on the plasma membrane, and only 5% of total cholesterol is located in the endoplasmic reticulum. So, which means that their lipid composition is so different, and lipid organization should be very different as well. Okay, to target that, first we selected Nile Red. It's our preferred dye, and now we considered we could label specifically different organelles, and for that we just add a specific targeting group, like we did for plasma membrane, to target using our amphiphilic anchor. For lysosome, we use a special group that will be protonated and accumulate in lysosome, then hydrophobic group for lipid droplets, then charged group for mitochondria, then different reactive groups to target ER, the plasmic reticulum, and a lipophilic chain to target Golgi. And we were lucky, all these molecules worked well. So plasma membrane you have already seen. So here colocalization of the dye with the reference marker of membranes. Then for ER, it colocalized well with the ER markers, both ER probes. Then for mitochondria, it colocalized with mitotracker, and the lyser with lysotracker, and then the same for Golgi, we had a good colocalization, and for lipid droplets. So we validated each probe with the corresponding reference, and then we went to image the polarity and lipid organization. So for that, what we do again two-color imaging, and then we construct a ratio. And when we constructed the ratio and put them on the same scale, we was very happy to see that in each case, the color was characteristic for each organ. But the interpretation of this result is not that obvious, because the probes are a bit different. So to be more independent of the probe structure, what we did, we calibrated each individual probe using liposomes. So in case of, in each case, we use DOPC liposomes or sphingomyelin cholesterol liposomes, and did the image, and like this constructed the color. And then this could be used to interpret the color that we observe here. For each case, we did the same, and you can see that their response to lipid vesicles is a bit different for each probe. But then we can use this to interpret the results. For lipid droplets, it was different, we needed oil. And then we introduced a quantitative parameter of, of lipid order parameter, where we relate the color to these two. And then depend on whether this color is closer to this one or to this one, we can conclude whether it is rather ordered or disordered. And based on this, we could clearly conclude that the most ordered phase is observed, ordered lipids are observed in plasma membranes, and then you have mitochondria and then all others. The most disordered were Golgi and a certain type of ER. In fact, depend on the probe, the order was still different because it labels different parts of proteins actually. These two probes. So here, depends on the probe, you will see the different order. So now to see whether we are really on the membrane, we extracted cholesterol. So cholesterol directly addresses lipid composition, and so we could see whether the probes are sensitive to lipid to extraction of cholesterol. Of course, for plasma membrane probe, no surprise, removal of cholesterol decreases lipid order. But the effects were a bit different, but they were observed for all markers. We could see changes in lipid order. And the strongest changes were in plasma membrane in the mitochondria, but they were observed for particularly all. And then when we added cholesterol, the effects were opposite. So the effects on plasma membrane were rather weak and on mitochondria as well, but they were much stronger for all other organelles, and especially for lysosomes here and Golgi, and these are the organelles which lack cholesterol. So the addition of cholesterol influenced more these organelles. And then we say, okay, can we follow some kind of stress parameters which could affect lipid composition? For example, lipid oxidation by using an oxidative stress with a hydrogen peroxide. And so what we could see that all these organelle-specific probes reacted very differently, changed very differently their color on oxidative stress. Plasma membrane a little bit, lipid droplets a lot, they were quite sensitive. Then lysosomes were also quite sensitive. Golgi and mitochondria also reacted quite strongly. And so this reflects actually oxidation of lipids, and those oxidized lipids, they produce effect on polarity, local polarity. And another parameter that we wanted to check was their mechanical stress, and that now it's a popular approach to produce a hyperosmotic shock where by changing the salt concentration, the cell shrinks, and this produces their mechanical stress on membranes. And we could observe for plasma membrane very small effects, but we observed heterogeneity on the membrane. That was important. And then the most affected ones were lysosomes, where we observed quite strong changes, and then mitochondria, and on one of the two ER probes. And so the mechanical stress could change either interaction between membranes, one membrane and another, or protein membrane interaction, in this case probably more affected by mechanical stress, or directly lipid membrane properties were affected by the mechanical stress. Okay, so I come to the conclusions. So I showed you several stories where we use solvatochromic molecules to address lipid order specifically at different parts of the cell, in particular on cell plasma membrane. So the initial probe that we developed helped us already to identify changes in lipid order on the whole cell. Then using super resolution imaging, we could by using low affinity probes, we could go to decipher with higher special resolution lipid organization on the cell surface. Low-down-based design probe could be very interesting to do imaging of lipid order, especially with different fluorescent proteins, because it has blue color compatible with green and red colors. And then the organelle-specific probe, lipidic probes, based on Nile Red, now we can address properties of polarity lipid order in each specific organelles to follow lipid organization changes like oxidative stress changes, mechanical stress changes, etc. And so I would like to thank, of course, the group which worked on this field a lot, especially Dmitro Danilchuk, who actually developed many of the probes of the present talk. Then Mayor, who worked with me on different sorts of probes as well, and other researchers, and also their fellows who worked with me quite some time ago, Alexander Kucherak, who in 2010 actually synthesized this NF12S probe. Then Biophotonics group with whom we collaborate on some microscopy applications. And of course, collaborators, external collaborators, Kate Su from US and Eldin Sezgin from Sweden, now before it was in UK when we did these experiments. And so I would like also to thank a lot of different support, financial support, especially European Research Council, who helped me to build this group and have instruments and have really time for this for science. And I thank you for your attention, and I will be really happy to have questions. Thank you very much.
Thanks a lot, Andrey. Fantastic. It was a really good tour for us from NR12S all the way to organelle-specific ones. So there are already many questions in the chat, but first I want to ask one question that it always puzzles me. You always talk about spectral shift, right? So a lot of people also use lifetime shift. When you think about the design of the probe, shouldn't they go exactly hand in hand? Or in the design, there are something that you can change that would make lifetime more sensitive or the spectra more sensitive?
Yeah, it's a very good point. So lifetime imaging, some people prefer lifetime imaging because it's a one color base, so you don't have chromatic aberration problem. And so I understand that some people prefer lifetime imaging system, although personally I prefer color because you need less photons. Certainly less photons, but the probes are not the same. The probes cannot be the same because color change is not necessarily translated into their lifetime change. So what I could say about solvatochromic probes, from our experience, you can get out from solvatochromic probes a robust lifetime response when you position your detection at the blue edge. So this was shown by others. But other labs, but we did it for pyrene probe, which was a rock stable molecule which didn't change lifetime at all. And when we recorded a whole spectrum, there was no lifetime change. And then we say, okay, you have this so-called solvent relaxation process. And so this means the band is shifting towards the red. So when you place at the blue part, you will see actually the loss of more rapid decrease of more rapid decay when the solvent relaxation is fast. And indeed, when you place the blue edge of solvatochromic probe, you will observe nice response to lifetime for practically any solvatochromic dye. But now, if we talk about specific design of the probe, yes, you need to design a probe which is badly emissive. Actually, for that, you need a kind of flexible molecule, which is, for example, molecular rotor like probe, or you have push-pull molecules which are very flexible. But in this case, you need to do a compromise because when you do that, you decrease photostability of the molecule. You also may increase phototoxicity of the molecule. And of course, you decrease brightness and you hope that in ordered phase, you gain 100% quantum yield, but it's not always like that. That's why I would say, it's probably better to either have this pure molecular rotor system, but really work for 100% quantum yield in ordered phases, and then these ordered phases will drop, or to go for really pure solvatochromic based probe with high quantum yield and high photostability and low toxicity. That is the problem. The probes, they are phototoxic frequently, especially those environment sensitive probes, because you have lots of charge transfer, etc. happening. Okay.
Thanks, Andrey. So I will go to audience questions. I just remind again, this talk, including Q&A session, will be recorded and published in YouTube. So if you have some sensitive thing in questions, please keep this in mind. So the first question is by Rafael Lira, and he's asking, is it possible to decouple information about medium polarity from viscosity and order, for example, using solvatochromic dyes or molecular rotors?
The medium polarity means polarity outside the membrane? Or?
I guess it's the solvent, yeah.
Because the thing is that when you use molecules, especially solvatochromic molecules, they are not very emissive in the medium. So they, moreover, we try to use targeting groups which has efficient binding to the membrane. So they are usually amphiphilic or hydrophobic molecules. So therefore, there is a minimum emission from the medium. But I would say it is possible to have a probe which would be redistributed in the medium and in the membrane, or only in the medium and not in the membrane. Technically, it's possible, but you will always have less light from the medium for multiple reasons. On one hand, in solvatochromic molecules, they are less emissive in polar media, in the water, but also you do not have effect of concentration. Because when it's on the membrane, the molecules concentrate in small space, you have a lot of light. But in the medium, you have all of them are diluted, so you have less light. And so, but in principle, it is possible to do.
Okay, so Ed Lyman wants to ask a question. Ed, could you please unmute yourself and ask a question?
Yeah, I didn't want to type the whole thing out. Sorry, I'm lazy.
Sure, go ahead.
Yeah, that's fine.
Follow up to the previous one, actually. So, you know, we've worked a little bit on trying to quantitatively model Loredan emission and using quantum mechanics and molecular simulation. One of the things we learned in that project is that Loredan emission in bulk solvents depends not just on polarity, which is kind of an average property, but also molecular interactions, in particular the hydrogen bond.
Exactly.
Whether the solvent can donate hydrogen bonds or not. And so I wonder, you know, is there a danger in making a one-to-one correspondence between the spectral shift and order? Because it occurs to me that there are probably other molecular interactions. A lot of these things have delocalized pi orbitals. You bring a cation in, you know, I don't think of that as polarity either. So I'm wondering, you know, how much dirty laundry is there that we haven't thought about?
It's a really nice question. In fact, it was a headache of my PhD thesis, let's say. When I worked on that, it was very much for me as a chemist. I imagined that Loredan and all molecules containing carbonyl group, the hydrogen bonding will play a key role. And in fact, hydrogen bonding corresponds to almost 50% of solvatochromic shift when you compare to a dielectric effect. So this means that hydrogen bonding plays a huge role in the response of the probe. But the thing is that all these interactions are transient, so you do not have very clearly defined species. When you, and so if you try to play and do the deconvolution of this spectra, you will come up with 10 different bands in the band of Loredan. It is so complex. You can have so many different species because so many sorts of interactions, orientations, etc. And another issue, and especially it concerns Loredan, and this was published already quite far ago, I think in 2006. I think we observed a so-called bimodal distribution of the molecule, which contain carbonyl, because you can consider molecule which is bound with hydrogen bond and not. And those which are not bound, they're deeper in the membrane. We observed that for 3-hydroxyflavones, but when we look the spectra of Loredan, we noticed also a kind of bimodal distribution of those molecules. And this means that if you want to use those probes, it's better to fix them on the membrane and to not allow them to distribute too much in the membrane, because they could be either bound or not to water. And that would create very complicated spectroscopy. And so at the end, to simplify all that, we cut the spectrum into two and we divide the two, and then analysis is more or less, as you said, the averaging. And if you go into detail, it's a hard life, especially for the molecules which are not having precise position in the membrane and precise orientation, because Loredan you can imagine like this and like this. And you can imagine it in different places in the membrane, bound or not to water. So many possibilities. And for Nile Red, for example, we started with fixing with a linker, etc. And the funny thing is that if you place properly, the fluorophore will have a strong response to the order. And if you place it differently, you won't have any response, depending on where you position the fluorophore.
Ed, you have another question? Is that correct?
Yeah, I can ask it real quickly. So the
Yeah.
MBD experiment was really cool. When you had the organelles stained and you hit the outer leaflet of the plasma membrane with methyl beta cyclodextrin, but there must be a lag, right? Because you're only depleting cholesterol from the outer leaflet. And so changes in emission that you see from the organelles must occur after cholesterol redistribution.
Yeah.
But before the cell can compensate.
Yeah, in fact, you know, we are here. We were as a chemist. So what we did, we just come to kind of saturation conditions. We applied for sufficiently long time this extraction process to ensure that we are depleting everything. So we deplete the cholesterol at the membrane, and then of course, then there is a redistribution of cholesterol, and slowly we get to depletion of the whole system. And in fact, if you do it for short time, it is plasma membrane that reacts. But that needs a much more precise study, and this is what we didn't do. So the fellow who did the experiments, he already had to synthesize, characterize all these probes at the same time, and can do all experiments in parallel. It was completely crazy. I think this has to be done with one probe, for example, on a membrane, and one in mitochondria or in another place, and then do a very precise time effect, cholesterol extraction, and then recovery of cholesterol as well. So all this can be done in principle.
I think it would be very interesting, right? I mean, there's not a lot of detailed quantitative information about, you know, how long these things take and how they happen. And
yeah, I totally agree. And in this case, you can follow the dynamics of cholesterol redistribution in the cell. Transport, it doesn't take 1 second, neither 1 minute. It's relatively slow for cholesterol. Yeah.
Thanks, Ed. Thanks, Andrey. So our next question is by Johannes Morstein, and he is asking, so first of all, very interesting talk. Thank you. Have you also looked at subcellular localization of longer chain Lordan derivatives like PATMAN? I imagine they could also be enriched at the plasma membrane.
Yeah, PATMAN, yes, a nice molecule. In fact, I guess it was synthesized with the idea to have more specific staining, but it is a cationic molecule. As I remember, it is a cationic molecule. It has a single cationic group. And in this case, molecule tends to very rapidly to go to the inner leaflet because of transmembrane potential. And then they will have all chance to accumulate in mitochondria. But in any case, once it's in the inner leaflets, it will start redistributing everywhere. And that is the limitation. Unfortunately, I have not tried PATMAN, but I also did not see so many cellular experiments using PATMAN. And I think the reason is probably that it doesn't have characteristic binding of a given organelle or plasma membrane. Yeah.
Thanks, Andrey. So there are two related questions by Betul and Christina. So they're asking how your probes can first be influenced by the membrane potential, ion channel activity. Second, how they can influence the membrane potential.
Okay. So, but in fact, the response to membrane potential of these probes is actually was studied by a group who took from me the NR12S, and it's a group in Switzerland. I don't remember the name, but so these fellows, what they did, they actually wanted to, as it is a solvatochromic molecule, it is supposed to be sensitive to electric fields. And so it's so-called Stark effect, and so the dipole interaction with the field. And NR12S was surprisingly good in detecting membrane potential. And but it's funny because I did a lot of work during my PhD to develop a probe for membrane potential, and it was not good, the one that we got. And now I'm doing probes for lipid order, and then they turn out to be membrane potential probe. Okay. Anyway, so it indeed works, but this probe, NR12S, was not really designed to be perfect membrane potential probe because it is not perfectly vertical. But in cholesterol-rich membranes, it is relatively vertically oriented. So it can detect membrane potential. It can detect dipole potential as well. So which is this one between the middle of the membrane and the surface, because it's oriented vertically again. And how it can influence, of course, it can influence membrane potential, especially dipole potential, because it's a dipole. But in the ground state, it's not so dipolar. So it's more dipolar in the excited state. Moreover, we use low concentration of probes. In fact, what we like very much is to add really nanomolar, a few nanomolar of this probe. And if I go back to the earlier applications of TNFs probe, usually advised concentration is micromolar, 1 or 2 micromolar. And here we are using 10 nanomolar. So we already more or less respecting the ratio of probe to lipid 1 to 100, which is supposed to be for membranes, for lipid membrane. So I do not think 1% of the probe can really change significantly membrane potential. Anyway, as soon as it doesn't make holes in the membrane, transmembrane potential will remain there, but dipole potential I think will remain the same.
So related to this, Andrey, you know, we also in collaboration with you, we really want to dissect what the probes are really probing, right? So if there is, can we get clean lipid order, but no electrochromism completely? So for
lipid, yeah.
So we know this, the ANEP dye, for instance, we published with Martin Hoff that it's definitely mixed. We cannot really get a clear answer from ANEP probes. Lordan, probably a bit cleaner. Do you have any feeling about Nile Red? I know we are doing the experiments at the moment, but do you have any feelings?
I think originally ANEP probe was designed for detecting membrane potential. And in fact, it has a really ideal structure. It's a very long dipole, perfectly, supposed to be perfectly vertically oriented in the membrane. And this probe would be the most sensitive to the electrostatics of the membrane. Nile Red is already weaker than Prodan, Lordan will be even weaker. So in this respect, I would say it will be something intermediate, probably between Lordan and ANEP's probes, because what I would say, what actually detects ANEP probes and Lordan and DNF. In fact, you have a bunch of combination of effects, because on one hand you have polar species like water present, you have hydrogen bonding species present, and then you have a dynamics of the membrane. And all this combination of these interactions influences the spectral shift. So in fact, for me, already between Nile Red and Lordan, there is a huge difference in the effect of cholesterol. We know for sure that Nile Red is extremely sensitive to a combination of cholesterol and sphingomyelin. But it is not so sensitive to the presence of cholesterol in disordered phase. It is sensitive, but not that much. But Lordan and also Pro-12A is crazily sensitive to cholesterol in all phases. The ordered phase, disordered phases. So there is something quite fundamentally different in sensitivity of these probes. For ANEPs, it's a bit different. I think it's something intermediate. It is sensitive to cholesterol also for all membranes. Yeah, so I would say electrostatics will play a role in all of them. And Nile Red should be closer probably to Prodan Lordan, but still you cannot exclude electrostatics effect.
Thanks, Andrey. So next question is by Devrimöz Aslan, and she's asking if you tried ER stress when you stressed with the intracellular probes with mechanical stress and
I think we already started that. So we did this experiment with application of hyperosmotic shock, where we squeeze the cell. And in this case, I think we already imposed some mechanical stress. And there were, for example, we had recently a question about it, where people said to me that it's possible that because of this, you impose the interaction of ER with plasma membrane, for example. And this would start changing the behavior properties of the ER locally. And because you start forming really very close points of interaction. So, but we did not characterize that in detail. I think if you are interested, you can always try, but I think it should work.
Thanks, Andrey. Next question is by Irene Alvarez Guerra. Thank you for the nice talk. I'm wondering if the internalization of the different probes could be hindered in cells with cell wall, like in yeast.
Okay. So the question is if a certain type of membranes would not allow their organelle probes to penetrate, right, to get inside. So first of all, for plasma membrane probe, we have never seen this probe. So plasma membrane probe stain membranes of all type of living cells. So you have bacteria worked very well, plant worked surprisingly well, and of course mammalian cell cultures worked very well. So I think the cell wall do not disturb binding to the surface. Now the binding inside, it's probably would happen rather because of if the cell wall is charged, and for example our mitochondria probe would bind to cell wall because it's charged. But otherwise, I don't think the pores are relatively large, and so those probes go through. So I don't think there should be a problem. But we did not test, for example, those organelle probes for the moment were tested only in mammalian cells.
Thanks, Andrey. Next question. I think I can even answer this question. How open are you to share or the published molecules? I can say Andrey is very generous, or alternatively, are they commercially available?
Yeah, so I can tell you, yes. So I am very generous until I will have some obligations with the provider. But for the moment, for Nile Red probes, they are still totally free of charge available from our lab. And so I send them. It's only problem that sometimes I'm slow because I have too many demands and I have to do science and not posting them every day. And so I usually collect many demands and then I send at the same time. So sorry if I delay sending, but we do it for free for the moment. We just distribute. There is no any special condition for that. But on the other hand, what is happening now is that NR4A and NR12A will be commercialized really quite soon, within a few, probably with 1 month or two, by cytoskeleton. And for organelle probes, still not there. It still will be available. We will distribute them. But I think they will be also commercialized one day. But I think it's better because like this, you will have quality control ensured. And yeah, so it should be done one day.
Sounds great. Thanks so much, Andrey. I think this is the whole questions by the audience. So maybe I can ask one last question, a bit unrelated. So you said you developed this quencher. Can we develop quencher for any fluorescent probes?
Technically, yes, because I think Viologen is quite universal because the quenching by electron transfer usually requires a good match between redox potential of the fluorophore and the quencher. But Viologen is so strong electron acceptor, so I think it will quench practically anything. So therefore, I would say this one should be quite universal.
Okay, so I will try this. We want to quench, for instance, body P.
Yeah.
Or NP. Yeah.
In many cases, a general trend, it will be stronger for quenching molecules which are neutral or negatively charged.
Okay.
Probably it will be a little bit less powerful with cyanines and rhodamines because they're cationic, so they will be less happy to give the electron. But I would say it's better to try. But for example, sodium dithionite is not at all universal bleacher. It bleaches NBD and it bleaches Nile Red, but it doesn't bleach other dyes. Maybe it can only decrease their fluorescence, but not, for instance, for Prodan, we could not. And we actually spend a lot of time trying to bleach or quench Prodan or Lardon. It was very difficult, but this guy, Viologen was very efficient.
Okay, so I will definitely try and let you know if it works.
Yeah, it would be interesting to see how universal is this question.
Yeah, sounds great. Thanks so much, Andrey. That was amazing. Thanks for
Yeah.
this amazing tour.
Thank you very much.
And thanks again for joining us today again.
Yeah, thank you. Then bye bye, everyone.
Thanks.
Thanks.